In this video, we'll be looking at the qualitative testing for biological molecules, the glucose Benedict's reagent practical, method 1 and method 2. Here we have the chemicals that we need to be aware of, mainly hydrochloric acid, which might cause skin or eye irritation. Health and safety below, pause the video and read. Here we have the list of equipment that you can find for this experiment. Mainly acquiring the different types of samples, 1% glucose, sucrose, protein, and also distilled water, but not necessarily in that order. We will be figuring that out later. Here, just some photos going through what some of your apparatus should look like and how you might want it set up. So we have the Benedicts, the pipettes, your test tube stands and your test tubes or boiling tubes. Here we have some small syringes. Here we have our 4 different concentration solutions. We don't know which is which, we need to figure that out. And finally, your water bath. Now, our reagents include, we have hydrochloric acid, sodium hydrogen carbonate, and we have sort of sorted labels, pencils, and tweezers and spatulas at the end. Here we have method 1, testing for reducing sugar. Please read all the steps at least once before attempting to start. Now, at the beginning, you will need to get your 4 test tubes and label them A, B, C, and D, as I have done here. Now, before you do the same mistake that I did, we are not going to add our Benedicts first, we need to add our sample first. So I had kept mine to the side, as you can see on the right hand side of your screen, and I switched out for my other test tubes. When gathering your samples, if your syringe does not reach the bottom of your sample tube, just tilt your sample tube slightly to the side, and that should be alright. Be ever so careful when drawing up your sample into your syringe. It's better to have a little bit more than less, because you can always expel until you get to that 4 centimeter cubed mark. You are going to complete that same step for A, B, C, and D. When you are putting your sample into your boiling tube, just be very so careful not to expel it at high pressure, because that might cause some of it to come out of your test tube. Just do it ever so gently, and that should be more than fine. You would need to create a results table for observations in between each of your steps. when conducting your sample and your water bath steps. Make sure that is ready before you add your Benedicts. As soon as you add your Benedicts, note down any observations you see. If there's no reactions, write no reaction. Then you put it in for 2 minutes. Here you can see after 2 minutes what my samples look like. You take them out, you note down any observations, the color changes, and then you put them back in for a further 2 minutes. So method 1 will have 4 minutes in total in your water bath. At the end, you leave them to cool just for a second, and you can put them in your test tube stand and compare them against each other to sort of see how they are and note down what observations you see. Now, we move on to method 2 for a non-reducing sugar, and we are going to complete the first steps exactly the same. We are going to put our samples in our boiling tubes and label them A, B, C, and D. We are going to place all our boiling tubes in the water bath and incubate for 2 minutes once we have added in 2 centimeters cubed of 2 mole decimeter cubed of hydrochloric acid to each of our samples. Once you have done that for 2 minutes, you will take them out and allow them to cool. Next, you need to neutralize the acid, and this is the tricky part, by carefully adding sodium hydrogen carbonate to each sample until no more effervescence or bubbling is observed. Now for me, sample A bubbled quite aggressively. I added a little bit more, and you can sort of see how much bubbles were produced. I let that sit and rest for a second to get some of the bubbles to come down a little bit before moving on to sample B. Sample B, you add a little bit once again until you observe no bubbles. Now, for me, some of my powder remained on the side of the test tube, so if that happens to you, it might be difficult to drop it directly into your liquid and not touch the sides. Just tilt your test tube ever so slightly. That's what I'm doing sort of off camera. You can sort of see now that there is ever so slightly just a little bit of effervescence still observed, so I'm going to add a pinch more for sample B. Give it a little swirl carefully to sort of see if there is still any reaction going on. Just moving it to the side again to get some of the powder that was stuck at the upper end of the test tube. I can see there is still a slight reaction going on. So just trying to get all of the sodium hydrogen carbonate that's along the sides to see if that will now stop reacting, which it seems to be finished. You are going to do this for samples C and D as well before moving on to the next step. Now, once you finish adding your sodium hydrocarbonate, you need to check the pH of your samples by using indicator paper. So you're going to take your tweezers and grab a small strip of indicator paper and drop it into your samples. You are going to use your accompanying pH card to read what the indicator is at. Most of them should be at 7. You get your measure out your Benedict's reagent 2 centimeter cube and add that into your 4 samples. Now, if you find yourself a bit slow on time, just like how I was, what I did was I prepared my samples for method 1 and method 2 and then put them simultaneously into the water bath, or very close one after another. And here you can see my samples at the 2-minute mark for method 2, and I will do the same here. I have my samples for method 1 that are ready. They go in for the amount of time that is specified in the method, and then make sure you are updating your results table as you go. Then at the end you should have 2 sets of samples, one from method 1, one from method 2, and then looking at your results table and your observations, you should be able to try and figure out which of your samples was which of your different solutions. For me, my sample A was my protein, my sample B was my sucrose, sample C was my water, sample D was my glucose, based on the observations that the reactions the samples had with my 2 different reagents.